will Paul Wall.
Cells move. Everything in your body is made of cells, and all of those cells are constantly moving, growing, and dividing to create more of themselves. So they're dynamic. Now we need a way of tracking the progress of those movements. So I want you all to raise your hand if you've ever looked into a microscope and seen something like that. Now I bet none of you have looked into a microscope and seen something like this. Because these cells are moving at such slow speeds, we can only track the movements by using time-lapse imaging, which is taking images at set intervals over a duration of time. We actually made this video using time-lapse imaging down in our lab. Now another aspect is fluorescence imaging, which is staining cells with certain fluorescent probes, allowing us to see cell markers to use for advanced cell research.
Now to see these cells, especially with fluorescence, you need something called a live cell imaging system. Now there are two cons with obtaining and using said system. The first is in the market today, these systems can cost upwards of $40,000, which for smaller institutions like ours, as well as independent researchers, this can be out of budget. The second con is, as you can see in the picture, the overall system is very big and bulky. It is a lot of pieces of equipment working together to make up the whole system. Now, in literature today, there is only one published paper that has successfully created an economical live cell imaging system. Their components include an on-stage incubator, temperature control, using a cell phone for imaging, as well as shutter to prevent damage from bright field light. Now, what they did not include, and what our team set out to include in our specific project, was using fluorescence, motorized stage control, and CO2 control.
The objective for our project was to create an affordable live cell imaging system, particularly one that will interface with the current inverted fluorescence microscope available in our laboratory. This system will incorporate an incubation chamber that is humidity, temperature, and CO2 controlled, automated stage and shutter control to allow stage movement and shutter function, and then remote monitoring and imaging with our system.
For the research and design criteria for our project, we split up our components into 4 separate modules. For the first module, we wanted to design the incubation chamber out of a material that could withstand the environmental conditions of the human body in terms of temperature, CO2, and humidity. This can be regulated by our second module, which is our temperature and CO2 control unit. Our third module is our imaging system, and we wanted to be able to image for up to 24 hours. And finally, our last module, which is our stage control. We wanted to be able to image multiple wells in the well plate, as well as have a shutter control for our fluorescent light regulation.
Now moving into our design constraints. In terms of time, we were both restricted by the deadline set by the project itself, as well as our limitations in times that we had access to the lab, being as if that was a shared space. Additionally, in terms of budget, we are limited by our funding, which was $1,900, as well as our own economical goals for the project, being as our end goal was to make the system more affordable than current commercial models. Additionally, in terms of compatibility with existing equipment, we needed to ensure that our system worked with the inverted fluorescence microscope located in the lab, as that is what we had access to interface our system with.
So then this is our final design. This design consists of a heating and CO2 unit, which is indicated by number 1, it's a styrofoam box behind it, which is then connected via a tube to our incubation chamber designated by number 2, which sits on the microscope stage. Number 3 is our imaging system, which sits within the microscope lens so that we can take images and collect data. And lastly, our shutter and stage control. The stage control, which is designated by number 4, controls our stage and our shutter. Now we'll be going into more modules.
I will now be speaking about our incubation chamber. So for our first and alternative design, we have our box and window material made all out of polycarbonate. Indicated in the middle is the cell tray where the well plate would sit. Surrounding the cell tray is a silicone gasket to ensure when the lid is placed on, proper insulation would be present. Now the reason we did not go with this design is because polycarbonate material does not allow fluorescence light to pass through. So, in our final design, we replaced the base and lid with glass to ensure that the fluorescence light would pass through, and so our images would be clear. Our incubation chamber is properly sealed using epoxy to pull all of the components together, and also adding an extra layer of silicone to make sure there are no gaps, and to make sure that there is no airflow outside of where we want it to be. The lid, because we need access to the cells to perform things like assays, we sealed it with parafilm, electrical tape, and Velcro for easy access. The inlet and outlet funnels, placed on the right and left-hand side of the incubation chamber, are epoxy to the chamber, as well as silicone around for extra circulation. And also, our sponges are placed on the opposite sides of the well plates for water control, which are submerged in water to also account for the humidity.
So now moving into a little bit more detail about our inlet and outlet funnels. These were designed custom made in a 3D modeling software called Fusion 360, and we did this to ensure proper airflow through our incubation chamber system. As you can see for the inlet, there's an additional cutout, and this is used to house our CO2, humidity, and temperature sensors, and these help to ensure that the air flowing through the system are at the proper conditions. And then afterwards, we sealed those in place with electrical tape.
Moving into our temperature and CO2 unit, this is our alternative design. Now this design utilized two large styrofoam containers, one of which held our sensors and the other of which held our heating unit. Now this design was a closed loop system, and while we did keep our final design being a closed loop system, we did not use this as the multiple boxes and the largeness of it resulted in too much heat loss and caused inaccuracy in our sensor readings.
Now we will talk about our final design for our temperature and CO2 control unit. So we designed this 3D model, which shows our fan-to-tube connection using the software Fusion 360. We integrated a fan into our heat system in order to circulate our temperature, humidity, and CO2. First, we embedded our fan into the styrofoam box, and then we added our fan-to-tube connection and secured it with 4 screws. Finally, we added our non-insulating tubing and sealed it with silicone tape. This allowed for us to create a continuous loop of our system.
Moving into how the system actually regulated heating and CO2, we had an Arduino Uno, which had multiple components attached to it, including a CO2 sensor, temperature and humidity sensor, solenoid valve, heat pads, and DC fan. Now how this actually worked was it used bang-bang algorithm, which means the program has a set range. And if the sensors read a value above that range, they tell the components to turn off. If it reads below, it tells them to turn back on. It's important to note that the range that we set it to is not what we want the internal environment to be. That is because having lower and more small gives us more consistent readings with our sensors.
The picture on the left shows our final temperature and CO2 system in its entirety. Indicated by the number 1 is our non-insulating tubing, and we kept this non-insulating to allow for heat to escape the system to avoid overheating. Indicated by the number 2 is our insulated tubing, which we insulated with styrofoam to allow for our optimal heat transfer into the system. The circuitry previously discussed is indicated by the number 3. Number 4 shows a thin, clear tube which allows for the entry of CO2 into the system, which is controlled and regulated by the solenoid valve. The image on the right shows the inside of our styrofoam container, which houses our heating pads. We attached our heating pads to sheets of thin galvanized steel in order to create optimal heat conduction.
For our imaging unit, our first design included using a smartphone camera which would be mounted to be directly into the microscope optics piece, and that would be controlled by a mobile application. However, the app software did not allow for remote imaging through the smartphone, so we went with our final design, which is a Raspberry Pi 4 mini computer. That was connected to a Raspberry Pi high-quality camera module inserted directly into the optics piece for stability. The imaging took place every 2 minutes over our 24-hour cycles, and those images were compiled into a video editing software called Kden Live to make the videos that you'll see later. This also performed system monitoring from our heating and CO2 unit. Then the Arduino data was uploaded directly into the Raspberry Pi in real time and uploaded to a CSV file for later processing.
For our shutter and stage control module, our original design consisted of a 3D printed gear which would articulate with a fine focus knob with the microscope, thus allowing us to bring the cells into focus inside the wells. X and Y function would be given by directly interfacing with the servos of the existing stage motor, and the manual shutter of the microscope would be used. However, for our final design, we decided to switch approaches so that all aspects of this component would be controlled similarly. That includes servo motors, which are controlled by a Raspberry Pi Pico W, and Lego scaffolding used due to its modularity and customizability to be able to fit our specific microscope. This allowed us to have limited movement in the X and Z directions. For our shutter, as you can see in the video, it is made of a servo motor and aluminum foil, which physically blocks the light. This is very important because the way fluorescent lighting works is that fluorescent light at a specific wavelength hits those fluorophores, then they fluoresce at another specific wavelength. However, if they are exposed to too much light, then they will lose their fluorescent capabilities in roughly half an hour. This shutter here, which we will see in a later video, allows us to image for up to 6 to 12, even 24 hours of fluorescent imaging, as opposed to half an hour without a shutter.
We will now be getting into our results and time-lapse videos of the cells that we tested. To begin, one of our 22-hour tests indicated this humidity chart right here. So as the graph you see, it's ranging between 35 to 40%. Now ideally, we wanted the humidity to remain high. Now I know you asked, well, high is not 35 to 40%. Although, we were just imaging for 22 hours, so the living environment of the cells was still perfect for us to view, so we accepted this data.
Next, we're going to go into our percent CO2 during incubation. We recorded this data for multiple hours. This only shows 1 hour to give you a better idea of that bang-bang algorithm I was talking about earlier. As you can see, the average here is 5.68% CO2, and that sits well within our range, and we have perfectly happy cells in this environment. Now moving into our temperature. Our temperature did sit at 36.05 degrees Celsius, which once again is perfectly within range, so we know our cells are healthy and happy.
Our first video is a scratch assay. So if you direct your attention to the cells on the left side, you can see that they're moving around and even dividing. Now the right side is open because a scratch assay involves making a scratch on the bottom of a well plate to create an opening between the cells. Now these cells don't move directly into that open area because they are so spread out and they have the ability to move in all directions, but you can see them moving and even dividing.
Our next time-lapse video is of contaminated fibroblasts. For this test, we purposely contaminated the culture dish to see how the cells would react. The small particles you see floating around are the bacteria. At the beginning of the video, you can see our fibroblasts are thriving, but towards the end, they begin to shrivel up and eventually pop, which indicates cell death.
In this last video, you can see fluorescently stained cells. The stain that we used was a general membrane stain, so basically it just stains the outside membrane of the cells. And this acted as our validation test to ensure that we were able to image successfully with fluorescence given our shutter function and our imaging, which we were able to.
So now moving into the conclusion, our incubation chamber as well as the temperature and CO2 control were able to successfully work together to maintain the proper environment for the cells for our experiment. In terms of imaging, we were able to get up to 24 hours of imaging through our system, as well as our system allowed us to remotely view our conditions such as CO2, temperature, and humidity. And lastly, we had a fully functional shutter control as well as a partial stage control.
For the impact of this project, we have created something that allows for novel experiments to happen here at Widener regarding live cell imaging. Student and faculty researchers can now do experiments such as looking at cell migration for wound healing, or seeing how cells interact in different environments and such things. We've also created accessibility in this science field because this is low cost and people can actually afford it, these small institutions and private research groups. And this opens opportunities up for the advancement of science. Now, to reiterate, a commercial unit could cost 40 grand. We did this for $1,400.
Now for some future directions, our first approach would be automating the video processing. So right now, the images are compiled but manually inserted into a video editor. Automating this entire video processing component would be a much better approach. And our second future direction is creating a mobile application for viewing. So once again, the real-time data is only available to the one user that is viewing Raspberry Pi. Creating an environment where anyone with access to the internet can view the real-time data such as the images and the conditions would be our next step.
Our final future direction would be to improve the stage control movement and precision. Currently, we do not have movement in the Y direction, and our X and Z direction movement requires more fine-tuning to be used for these longer-term experiments.
This concludes our presentation, but we wanted to give you something to take home with you. So if you take out your phones right now, you can actually scan this QR code, and this will take you to our team's webpage, where you'll have access to all 5 of our videos that we captured. Now we also wanted to give a huge shout out to Chris Hallahan. Without him, we couldn't have done some of the electronics that we did, and he just helped us out so much, so we wanted to thank him. Any questions?
So obviously you recommend this for useful for your fellow students and professors. Do you guys have a user's manual on it yet?
We're working on making our whole thing right now because we want to get published, so it's in the works.